Okay. Great. Okay, now I have to go back to sharing since I just left that. Okay, great. So I just started the recording, but you haven't missed anything. I was just reminding everyone that we're going to be focusing on this part of the viral life cycle, in which we focus on how the genome gets translated into the proteins that are required for replication of the entire viral particle. And if you remember, we're really basing this on a beautiful talk given by Britt Glaunsinger, who is a professor at UC Berkeley and an HHMI investigator. And once again, here's the link for the video on YouTube, but I've also uploaded it to B Courses. Before we get going with the, and return to the lecture by Britt Glaunsinger, what I thought would be helpful, you know, this is really challenging material, and so often it can be useful to hear the same material in different ways. So I thought I'd give a brief preview of what she's going to talk about, present it to you in a different way than she does, and then we'll come back and get into the you know, really the rigorous mechanistic approach that she's going to talk about. So for those of you who have previewed the video, and for those who haven't, the viral genome is about 30 kilobases long. And it actually has one very long open reading frame here. And this actually encodes a whole bunch of proteins. If I remember correctly, the entire genome encodes about 27 proteins, but for this slide, I just want to focus on this first very large open reading frame. You can see it's called ORF1AB. And to start, I'll just point out that this, this figure depicts the protein structures for each of the proteins that are encoded by this first open reading frame. And there are a few specific proteins that I want you to focus on. This papain-like proteinase here, which is called NSP3. The NSP is for non-structural protein. This 3C-like proteinase, and then we'll come back later to talk about this proofreading exoribonuclease. And the reason these two proteinases are important is because the virus has to conform to the rules of eukaryotic transcription and translation. And so what that means is eukaryotes cannot deal with a polycistronic transcript like this. They you know, typically in eukaryotes, and a single RNA is encoded into a single polypeptide or protein. And yet this virus needs this sequence to encode many different proteins. And so the way it does it, just to give you the cut to the chase, and then Britt Glaunsinger will explain it in more detail, the way it does it is that this transcript actually encodes these two proteinases. And the transcript actually gets translated as a single, what's called polyprotein. And then these proteinases go through and cleave between each of the, you know, each of the portions of the polyprotein that allow the release of an active protein. And so you can see this here. So this is a depiction of a related genome. This is the SARS-CoV from 2003. It's not CoV2, but it's close enough. And so what you can see here are the cleavage sites that are the target of the two proteinases, here and here. Okay, so these blue arrows are cleaved by this papain-like proteinase, and the teal ones are cleaved by the 3CL protease. Okay, and just a reminder, please feel free to, if there are questions, send them in through the chat function. Okay, so one question I had as I was preparing these slides was how are these two proteinases active before they're actually cleaved from the polyprotein? And unfortunately, I don't know the answer to that. So I'll just say I don't know. But it's something I'm sure that I can find out with more time, and I will try to get back to all of you about that. Okay, so there's one question. How did the proteinases release to cleave the long ORF? And is that question the same as the question I had? Okay, yes, the person asking is asking yes. So that is a question I don't have the answer to. I think it's very interesting. And without and I'm wildly speculating here, but I suspect that there's some low-level activity before, while it's still in the polyprotein, but I don't know the answer, and I will try to figure that out. Okay, so let me try to go forward. Okay, so those two polyproteins, those two proteinases cleave this polyprotein into these different smaller proteins that have activity. And one of those proteins that's extremely important for Cov2 is this RNA-directed RNA polymerase. Okay, because keep in mind, what's happened is that this long open reading frame ends in a stop codon. And so these proteins don't actually get translated off of the original viral genome. Kirsten, you're not muted. So.
Oh, sorry, pardon me.
That's all right. What happened? Okay, there we go. So what happens is that the RNA-directed RNA polymerase that's encoded by this long open reading frame actually starts RNA replication. So remember, this is a plus strand RNA ready to be translated. It replicates the minus strand, and this allows then for replication of the minus strand back into the plus strand, which then allows the expression of these. And you're going to learn about that in a lot more detail in the video, okay? So these proteins are the structural proteins. They include the spike protein that we already discussed, as well as that membrane matrix protein, a nucleoprotein that helps package the ribosomal genome, and then some other proteins whose functions are less well understood. Okay, so what I've also included in this slide deck are just a couple of other slides that sort of pull together a lot of what you're going to hear in the Glounsinger lecture. They are, to my mind, difficult to look at. There's a lot of information, so I'm not going to go through it right now, but I wanted you to have it as a reference that you can compare back to the video. And so what I was just very briefly summarizing here was the release of the genomic RNA into the cytoplasm, the translation of that first open reading frame, and we're going to talk about how you get two different polyproteins here. And then the release of these proteins, including the two proteases and the RNA-directed RNA polymerase that allow the replication of the viral genome into these two forms. And then we're going to focus on these subgenomic RNAs. So we're going to go into this in a lot more detail. I just wanted to give you a sneak preview.
Dr. King, someone has a question about, it's in the chat, but it says like, oh, is after here, maybe you can just read it.
Yeah, is that when the frame shift happens? Is that the one you're asking about? Yeah.
Mm-hmm.
So the frame shift happens in this long open reading frame. And so let me get, okay, it's here. So, so essentially there is a stop codon in the original frame of this long transcript. And then there's a really cool slippery, I'm trying to remember what it's called, slippery hexanucleotide that where the ribosome actually pauses, and then with some non-trivial level of frequency will actually frame shift, allowing these other genes to be translated. Okay, does that make sense? I'm looking at the chat. Hopefully that makes sense. Yeah, so this part is from the original plus strand genome that that came from the infectious viral particle. Okay, so let's, I'm just pointing out that I have these summary slides that I've included in the slide deck for you that you can refer to as you study. They all show the same thing, but with different graphics. And I've included the links for all of them so that if you find that useful, you can go and read more. But, you know, I don't expect you to know anything that isn't actually covered in the lecture today. Okay, so now I'm going to leave that. I'm going to stop sharing okay, new share. And now we're gonna go to the original lecture. So can you all see this slide that says acquisition of polybasic cleavage site? Let's see, is that sharing now? Oh, no, it went, sorry, it
says not Cov2.
Okay, shoot. That should be working now.
Yep.
Okay, good. All right. So I'm going to start this audio, and then as before, I will pause and pontificate and also try to answer questions that I see coming through. So here she's just talking about this polybasic cleavage site that we discussed briefly before, but she's gonna
sorry, could you because so you need to share the computer
audio.
audio.
Thank you. Good. Sorry about that. You'd think I would remember where that control is. Hold on. Oh, how funny. I had it on Friday or on Wednesday. Abby, can you remind me where the share audio is? Let's see. Sorry, everybody. Huh, I'm not finding it today.
If any of the students remember how to share audio, that'd be great.
Let me ask my 13-year-old son. He's 14 now. Nady, I don't remember how to share audio on the computer. Yeah, I thought it was in the advanced settings also. Here, he got it. Okay, thanks, Nate. This is the good part of shelter at home. You have technical help right there. Okay, sorry about that. Hopefully.
And it's particularly important because insertion of the polybasic site in other viruses has been shown to increase transmissibility, particularly for pathogenic influenza viruses. So it's going to be important to figure out whether the same is true for SARS-CoV-2. Okay, excuse me. So that covers entry, and we're now going to move on and talk about what happens to the viral genome once it has moved in to the cytoplasm of the cell. Well, the 2019 Cov2 genome has been annotated, and depending on the annotation that you look at, it's thought to possess about 14 open reading frames, encoding an estimated 27 or so proteins.
Now, one thing I want to point out that I found confusing when I first looked at this slide is that the proteins encoded by this open reading frame here are depicted below, but it actually wraps back around. So this is a continuous polypeptide. I hope that makes sense. So it's not actually in two pieces. It just wraps around and is continuous because it didn't fit the way they were scaling it.
Now let's think about this for a minute because it's kind of remarkable. Remember that the viral genome is a single stretch of RNA that is incredibly long. It's 30 kilobases long. But for any virus, the same is true for coronaviruses. Once that RNA is deposited into the cell, the ability to translate or generate proteins from that RNA requires the virus to basically follow the gene expression rules that are set by the host cell. And for eukaryotes, translation is a process that's generally a monocystronic one, which means that when the virus comes and when a ribosome comes and recognizes an RNA in the cell, it will translate generally one open reading frame, one gene from that RNA before recycling and falling off. This is different than prokaryotes, which of course have multi-cistronic RNAs where multiple proteins can be translated from the same RNA. Not generally true in eukaryotes. So how is it that from one RNA then, this virus is able to express 27 different proteins? Well, for coronaviruses, there are at least three solutions, or three well-known solutions, that the virus has evolved in order to solve this problem of expressing many proteins using the eukaryotic rules of gene expression and translation. And we're going to talk in some detail about those. The first.
Okay, I'm just going to interject. I think this is a really important point. The virus has to follow the eukaryotic rules. Okay, it's really depending upon the machinery that's in the eukaryotic cell. So, although a virus is not a eukaryote, it has to encode its genome in a way that can be interpreted by the machinery of the eukaryotic cell.
Is that, if you'll notice, a large portion of the genome is made up by a single open reading frame called open reading frame 1, which is separated into sort of two sub-open reading frames, 1A and 1B. This is a giant open reading frame that is basically translated into what's called a polyprotein. It's a series of many proteins fused together with no stop codons intervening them to generate one giant protein, which is then proteolytically processed, and we'll have that on the next slide. This protein is also generated in two different forms through the use of a programmed ribosome frame shifting event, which we'll also talk about on the next slide. That gets you translation of all of the open reading frame encoded proteins in that portion of the genome, which are generally the non-structural proteins of the genome. But it doesn't get you translation of all of the structural and other accessory proteins, which are found on the 3' half of the genome, the 3' end of the genome. And for these to be made, the virus uses a very unusual strategy of discontinuous transcription that produces something called subgenomic RNAs, and we'll discuss those as well. So to start, remember that the virus has to first make proteins that are going to be necessary for it to be able to copy its genome and transcribe the rest of its genes. And for any RNA virus, this requires an RNA-dependent RNA polymerase or an RDRP. If you are a plus-sense RNA virus, like coronaviruses are, your incoming genome is basically recognized as a messenger RNA. It's ribosome ready. So you don't have to package that RNA-dependent RNA polymerase complex or protein in your virion because it can be directly translated from the genome. And that is what happens. That's what is encoded by this giant open reading frame 1A or 1AB. So this is made, as I mentioned, into a huge polyprotein. Within this polyprotein are two proteases that the virus encodes. And these proteases, the job of those proteases is basically to now cleave this giant polyprotein, as shown here on the left, in the lower pullout into the individual proteins, which are going to have separate functions for viral gene expression and replication. And so you get proteolysis to generate many different proteins from one initially translated polyprotein. Additionally, you'll notice that this polyprotein, as I mentioned, is not just translated as one giant open reading frame to start. There's a frame shifting event. So a portion of the time, maybe 50 or 60% of the time, the ribosome will read through and there's a stop codon at the end of ORF1A, so it will stop there. However, the remaining percentage of the time, the virus enables the ribosome to actually read through that stop codon and continue translating down to generate a longer ORF1AB fusion. And that programmed translation read-through is a frame shifting event that is governed by two properties of the genomic RNA. The first is that right around that stop codon, there's something called a slippery sequence, and this is shown in the RNA diagram on the right. The sequence of UUU, AAA, AC. And when the ribosome lands on this site, it's known that it tends to have a propensity to occasionally slip back out of frame. Now, the frequency with which that frame shifting event occurs can be increased and is increased in these coronaviruses because just downstream of that slippery sequence is what's called an RNA pseudoknot structure. This is basically a highly stable RNA structure that causes the ribosome, when it encounters it, to pause. So the structure is thought to interact with the ribosome, causing the ribosome to pause over the slippery sequence, which increases the chances that it will slip back out of frame. If it slips back one nucleotide out of frame, that stop codon at the end of ORF1A is no longer read as a stop codon, and the ribosome can continue to translate through and generate the rest of the viral polyprotein.
So I'll just make an aside here. This is one of the things I love about biology. So this is a stochastic process. Not every, it's not binary. It's not like there's some on-off switch that says, yes, you're going to pause at this stop codon, X% of the time, and Y% of the time, you're always going to read through this stop codon, through this slippery heptanucleotide. It's really a kinetic phenomenon, and it just has to be good enough. So there's probably variability from cell to cell and from viroid to viroid, what the exact number of versions of PP1AB are translated versus PP1A. And as long as there's enough of the proteins that are encoded by this longer version of the polyprotein are made, then it all works out. Okay, let's see, there's a few questions. What's the name of the part of the genome that causes the ribosome to pause? It's right here, it's the stimulatory pseudoknot. And if the ribosome slips out of frame, how are the rest of the proteins translated correctly without having a frame shift mutation? So the idea is that all the proteins at this end, encoded by this end of the transcript, are encoded properly in the proper frame. There's then a stop codon. So if it doesn't slip, this part of the genome doesn't get translated at all. If it does slip to the -1 reading frame, then these do get translated. But of course, it could slip -2, and that would also be wrong. So it has to slip the right amount. Does that answer the second question for Mayuri? Okay, I hope it does. The slippery, okay, here's a question. The slippery sequence causes the frame shift, right? Yeah, well, yes, it increases the likelihood of the frame shift, the slippery heptanucleotide, sorry. This is the place where the ribosome can shift, and this is enhanced by the stimulatory pseudoknot. So Tess makes a clarifying comment, which is that the second set of proteins here in this polyprotein are encoded in the -1 frame. Okay, Are there any other questions before we move forward? Okay.
Okay. Then that protein is processed, as I mentioned, but how do you get production of all of the rest of the proteins that are found on the 3' end of the viral genome, the structural and accessory proteins? These are made from basically a nested set of what are called subgenomic RNAs that have, they're all 3' coterminal. So this is important if you think about it for how these are going to get their proteins expressed. These are not polyproteins, but by having this nested set of subgenomic RNAs, what this enables the virus to do is have each of these genes on the 3' end of the genome have a chance to be present as the 5' most open reading frame on a messenger RNA. So let's think about it this way, where if you are generating an RNA, for example, where in this case gene 2, which would represent the spike protein, for example, is at the 5' end, the ribosomes are going to come translate gene 2, and everything downstream of it, based on the eukaryotic rules of translation, is basically going to be viewed as UTR sequence, untranslated sequence. So only gene 2 will get translated into protein 2, or spike in this case. The same thing, if you generate a transcript in which now gene 3 has the chance of being the 5' open reading frame, that will get translated into protein, and everything downstream will be untranslated sequence. So every gene at the 3' end of every open reading frame at the 3' end of the viral genome has a chance to be the 5' open reading frame on a messenger RNA, allowing it to get translated. How this happens is quite fascinating, and involves another feature, which I hope you have noticed here on these RNAs that I've drawn, and that is that in addition to being 3' coterminal, all of them have the exact same sequence at the 5' end, and that exact same sequence is a sequence that is the same at the 5' end of the genomic RNA called the leader or L sequence. So how is it that you are able to get the same sequence, which is not present within this 3' end of the genome? How are you able to fuse that to the ends of each of these subgenomic RNAs? And that basically, the answer to that underlies how these are produced. It involves a series of sequences called transcription regulatory sequences, or TRSs, at the junction between each of those genes encoded by the virus, as well as at the 5' end of the genomic RNA, just downstream of the leader sequence, which is denoted in red here, are these conserved TRS sequences, these transcriptional regulatory sequences. And so as the polymerase is coming and copying the genome, it's going to reach these TRSs, which are at the 5' end of each of the genes. There's a core, highly conserved sequence within these TRSs. This is called the core sequence, or denoted as CS here in yellow. And so once the polymerase gets to these TRSs and copies this core sequence, it can either continue to copy, or it will now jump from that sequence, probably through a long-range RNA-RNA interaction, and base pair with the same core sequence that is part of the TRS at the 5' end of the genomic RNA that is just downstream of the leader, and then the polymerase will continue to transcribe, thereby capturing the leader sequence. So this looks something.
Okay, so that's a little complicated, but hopefully you get it. So it's not that those that this leader sequence is actually encoded throughout the genome. The key is that these TRSs are encoded throughout, and they can cause the newly replicated RNA to actually physically complement with the 5' end of the genome and lead to the replication of the leader sequence. Does that make sense? It's going to be explained
like this, where the nascent RNA is shown in red. The RNA polymerase starts to copy. You'll see that the TRSs are present at just upstream of each of the genes in the virus. As the polymerase gets to one of the TRSs, it either can read through that TRS and go on to the next one, or it will jump and translocate basically to the TRS at the extreme 5' end of the genome, finish its transcription to generate that fusion with the leader sequence. So this is discontinuous transcription. It allows for the generation then of a series of these subgenomic templates. Remember, these are copied from the plus sense RNA genome. So these are now negative sense or minus sense RNAs. They're complements of the genome, but not ribosome ready themselves. For that, the polymerase now has to go back, make copies of these minus sense subgenomic messenger RNA templates to generate the actual positive sense messenger RNAs that can be translated.
Okay, so a question came in. Does the virus particle contain a complete minus RNA genome and a plus RNA? Or I think is the first ORF positive RNA in nature and the 3' portion of the genome negative RNA? I'm not sure I 100% understand the question, but just remember that these are single-stranded RNA viruses. So the viral particle that infects the cell has a single-stranded RNA genome, and that is the plus strand. So that is the ribosome-ready version of the genome. The ribosome then starts to translate off of this genomic DNA. I wish I had a better slide up for showing you this. And then it's only after the RNA-dependent RNA polymerase is translated and starts to replicate the 3' end of the genome into a complementary copy that you start to get these structural genes translated here at the 3' end of the genome. Okay, so when the viral particle infects, it only has the plus strand RNA. It's in the host cell itself that that this part of the genome is replicated by the viral RDRP into minus stranded mRNA templates. And then the RDRP uses these templates to transcribe, to replicate the 5' the 3' portion of the genome here into these subgenomic mRNAs. Does that answer the question? Okay, yes, good. Okay, so another question is the short subgenomic mRNA templates come from the fusion of the RNA, and the longest ones are due to the continuous RNA replication, no fusion. No, that's not quite right. So what happens is the RDRP, the RNA-dependent polymerase, starts reading through, and remember when it hits these TRSs, sometimes it will just keep transcribing or replicating, and sometimes these sequences will instead complement with a TRS at the 5' end, leading to the addition of a leader sequence. Now, how does this happen physically? Remember that the RNA is probably folded up inside the cytoplasm. So even though in this linear version, it looks like this is really far away, it could be that in the cytoplasm, it's tightly folded, and so it would actually be more proximal. So the longest ones are due to continuous RNA replication. So imagine this is the longest one. It hits that TRS, and then the expectation is that it would then have this hybridization event here, or complementation, and lead to the addition of this leader sequence. Okay. All right. I'm going to keep going.
It's worth thinking about that this mechanism of discontinuous transcription means that there's a lot of polymerase jumping and probably facilitates what are known to be extraordinarily high recombination rates within coronaviruses, as high as I've heard estimates of about 25%. Most RNA viruses and plus sense RNA viruses have vanishingly low levels of recombination. And so this is a unique feature to coronaviruses, which may be interesting in regards to how they evolve, and perhaps also how they are able to maintain such enormous genomes. So this discontinuous transcription mechanism is quite complex and is orchestrated by a replicase that includes the polymerase, but many other proteins as well. And this replicase complex requires functional integration of the RNA polymerase, capping, and proofreading activities, as well as other things. And so what I'm showing you here on the left is a structure of basically what people think is the polymerase holoenzyme. This is made up of the NSP12 RNA-dependent RNA polymerase itself, together with two other non-structural proteins, NSP7 and NSP8, which are thought to help with processivity of the RDRP. As I mentioned, this is thought of as perhaps the core holoenzyme of the polymerase, and it is believed to be able to initiate de novo primer-independent RNA synthesis. In addition, the complex is associated through protein-protein interactions with another non-structural protein called NSP14, which is a bifunctional protein that has both capping activities and an exonuclease activity, which turns out to be a real paradigm-shifting activity for how scientists think about RNA virus evolution. I'm going to spend some time talking about that. But first to mention that it's not just this, but these proteins mentioned above, but in fact there are a variety of other viral processing proteins and activities associated with the replicase complex, not all of which are well biochemically understood, as well as sort of an undefined set, or at least an incompletely defined set of cellular proteins that may participate in its regulation as well. So a very complicated replicase complex involved in orchestrating this discontinuous transcription mechanism.
Okay, so can anyone tell me why the virus needs to have a capping enzyme for these RNAs that it's making from the 3' end of its genome? Any ideas? Yeah. Yeah, it needs to be capped. Remember, eukaryotic RNAs are capped. And so if it's not capped, it's going to be degraded. Okay, let me back up a little bit here.
Actions with another non-structural protein called NSP14, which is a bifunctional protein that has both capping activities and an exonuclease activity, which turns out to be a real paradigm-shifting activity for how scientists think about RNA virus evolution. I'm going to spend some time talking about that. But first to mention that it's not just this, but these proteins mentioned above, but in fact there are a variety of other viral processing proteins and activities associated with the replicase complex, not all of which are well biochemically understood, as well as sort of an undefined set, or at least an incompletely defined set of cellular proteins that may participate in its regulation as well. So a very complicated replicase complex involved in orchestrating this discontinuous transcription mechanism. All right, back to this exonuclease that I mentioned as being part of the polymerase complex. Turns out that the theoretical limit for how large an RNA virus genome can be is about 30 kilobases. And this theoretical limit comes from the observation that in RNA viruses, which all have RNA-dependent RNA polymerases, these RDRPs do not have proofreading capacity. So this is different than polymerases in our own cells. And this means that they are error-prone. And this error-prone capacity of RDRPs underlies the massive evolution that occurs during replication of many RNA viruses to generate things called quasi-species and mutant swarms that are highly characteristic of infections like HIV and influenza. And what it also means is that most viruses actually don't even come close to that theoretical limit of 30 kilobases. Most RNA viruses are in well below 20 kilobases, and most are in the sort of 10 to 12 perhaps kilobase order. Now, there are viruses, like, as I mentioned, coronaviruses and others in a larger grouping of similar viruses called nidovirales that have shockingly large RNA genomes, 30 kilobases. We even know some that are now beyond 30 kilobases, so even exceeding the theoretical threshold. And within these viruses, only these viruses, not all of them, but many of them, have this exonuclease activity that's present. And so this led to the idea that this exonuclease activity could actually be conferring a proofreading function on the RDRP, which, as I mentioned, was a real paradigm shift in thinking about how dependent RNA polymerases might actually be able to proofread. So indeed, in SARS coronavirus,
I want to pause for a second. Let's see. Uh-oh. Okay, good. So let's talk, because there was a question in Piazza about, now I'm trying to remember the exact details, but it was something about what does the exonuclease have to do with the size of the genome. So here's this correlation that we see that, there's a theoretical limit based on mutation rates, and then there's, and that theoretical limit is about 20 kb. And then there's the empirical observation that there are RNA viruses that have larger genomes, and the finding that many of these viruses encode this protein, XON, which has an exonuclease activity, which you're going to hear in a moment, helps with error repair. So I guess in the interest of time, I was going to try to draw out a discussion, but in the interest of time, I'll just say that the idea is that these viruses can't replicate with enough fidelity at the larger sizes unless they have error repair. So that seems not so confusing, just that as your genome gets larger, and depending, assuming that there's a certain error rate in the RDRPs, and it has a certain level of processivity, that you're going to need some sort of error repair to maintain the fidelity of the genome and some viability. I put in quotes because, of course, there's this controversy about whether viruses are alive, but if you at least want its genome to be heritable, there needs to be some fidelity in its replication. Now then that raises the question of why, if fidelity is important, why don't these smaller viruses also have this XON protein? And one interesting thing that we see in microorganisms, is that the synthesis of every protein requires an energy input. And so there's actually a fitness cost to every protein that's made, as well as a potential fitness benefit. And so it's a trade-off. And so what I suspect is that there has been selection against having an exonuclease in these smaller ones because they do okay without it. And they can, it's one less protein to make, and they can replicate more quickly without having to make this other protein that is not strictly essential for maintaining the fidelity of replication. Whereas for these larger ones, it is necessary, or at least provides some sort of fitness advantage.
Dependent RNA polymerases might actually be able to proofread. So Indeed, in SARS coronavirus, if the exon, this exonuclease gene is mutated, and then the number of substitutions or mutations that occur during replication of this virus are measured, you can see here from this graph that compared to the number of mutations that occur in the wild-type virus, there's more than a 20-fold jump in the mutational frequency in the virus lacking this exon activity. So you can see this spread across the rest of the coronavirus genome here, first focusing on the upper panel, where in dark, in black basically are the lines showing the frequency of mutation in the population during infection with the wild type virus, and the gray lines show the same thing during infection with the exon mutant virus. And you can see that there's a significant increase in the number and distribution of mutations that are acquired. This also renders these viruses mutation of exon renders these viruses hypersusceptible to mutagens, as shown in the lower panel, which include here what was tested as 5-fluorouracil. And so you can see, of course, 5-FU treatment, which is a mutagen, increases the mutational frequency of the wild-type virus, but further increases the mutational frequency, of course, of this exon mutant.
Okay, so it is 9:56. I'm going to just arbitrarily stop right here. Actually, the lecture becomes even more intense with this slide. So we're only responsible for the material up to the prior slide. But I, of course, invite you to watch the whole talk. I mean, I hope that you'll find this understanding of the biology of CoV2 really useful and interesting in the context of all of the public policy decisions that are being made, and thinking about this pandemic in terms of its biology as well as its implications for society. So I'm going to stop there and give people a chance to ask any final questions, and give me a chance to wish you all the best. It's been quite a roller coaster, but you've all been incredible. And so I just want to wish you all the best as you wrap up the semester and move on. Let's see. Thank you all for saying thank you. There was a question. Could knocking down or out the exon be a possible vaccine or treatment? Yeah, it would be a great target. And another great target would be, of course, the RNA-dependent RNA polymerase. So those are exactly the kinds of things I want you to be thinking about. If both what would be good targets for drugs, and also what would be good targets or good things to use as potential targets for vaccines. Is that the way? I'm terrible at saying that, Remdesivir works. I thought, yes, it does look promising. It seems to shorten the period of time that people are ill. It does not seem to improve. Oh, I don't know why it's still playing. It does not reduce mortality, it seems, but it does reduce the amount of time that people are sick. I thought I remembered that Remdesivir was targeting the RDRP, but please correct me if I'm wrong, and then it's actually targeting the exonuclease. Yes, someone says it shortens by about 4 days, and I think that's correct on average. Okay, so can I explain the subgenomic RNA transcription mechanism? So essentially what's happening, pardon me, is that if you remember correct, I'm going to go back to my original slides because I think it'll be easier to show you. Let me start. Okay, wait. Oh, my technical skills are collapsing at the last minute. Here we go. Okay, so here we go. So this slide, let's see if this one's better. they're all bad. I feel like they skip this important point. So essentially the RDRP initiates transcription, or sorry, replication from the 3' end. Can you all see this? Here, I'll put it in play. It initiates replication from this end, and then as it's replicating, it goes through these core sequences, these TRSBs, and with some frequency, that TRSB will cause that portion of the genome, here I'm trying to show you, that portion that's been replicated into an RNA to move over and copy that leader sequence. The leader sequence will then terminate.
I can't see your slides.
Oh, you can't see it. Ah, sorry, thank you. Can you see it now? No, wait, no, that's not. Oh, shoot, I'm so sorry. And in fact, Keynote just, you can't see this? Let's see. I'm trying to show you the genome of the Cov2.
Yeah, you might be sharing just the video and not your screen.
Yeah, it's interesting. As I'm trying to share it, it's not even showing. Okay, I'm just gonna try to explain it then. I'm gonna stop sharing and just try to explain it because it's not working. Okay, so hopefully we'll just use my hands and that will work. Okay, so as you have the RNA-dependent RNA polymerase, and I don't know if I'm being mirrored or not, so hopefully it's coming from the right direction, but the RNA-dependent RNA polymerase is copying the 3' end of the genome, and then it is it's hitting those TRS sequences, and those TRS sequences are then leading the RDRP to pull that shortened copy of the RNA over to the leader sequence, and then the leader sequence gets copied. And so what happens then is that I have to make sure I'm answering the right question. So then what happens is the RDRP copies the template back in the other direction. And so all you're doing is getting a copy of one part of the genome. So it's subgenomic. Okay, and so that new plus strand is from the 3' end of the genome is now able to direct translation of those structural proteins, including spike. Okay, are there multiple TRS sequences throughout to allow for different lengths? Yes, so there are TRS sequences at the 5' end of each of those structural genes. Okay. Good. So, so maybe I should pause there. I sent out an announcement with my office hours for next week. I have 2 office hours, I believe, one on Wednesday and one on Friday. Stay tuned for Professor Dernberg to provide details about the final exam. Give yourselves all a hug and take care of yourselves. I hope that you will look back on this time and just understand how amazing you've all been in this very challenging time. And I wish you a very calm summer and a fruitful fall. And you know, feel free. I always enjoy hearing back from people in the future about what you end up doing. So. Take care, everybody. Oh, I feel a little. Okay. Okay, I'm gonna say goodbye and wish you all the best. Okay.